First of all, I would like to convey our heartfelt thanks to Dr. Appu Rathinavelu for having kindly consented to deliver a talk today. I want to thank you on behalf of the organizers. Okay, Dr. Appu Rathinavelu is currently the Associate Dean, Institutional Planning and Faculty Development, and Executive Director, RumbaughGoodwin Institute for Cancer Research. Dr. Rathinavelu initiated his research efforts focusing on discovering new therapeutic targets and novel drugs for cancer treatment. He has published nearly 50 peer-reviewed research articles, co-authored a textbook, written 2 book chapters, and presented more than 75 scientific papers in national and international conferences. He also serves in the editorial board of several esteemed scientific journals and has conducted peer review of several scientific manuscripts for publications. Dr. Rathinavelu has received 4 patents and is currently planning to perform preclinical trials for his anti-angiogenic drugs with pharmaceutical companies. His primary research focuses on discovering novel methods for predicting treatment outcomes and developing drug combinations that will be useful for more potent and less toxic treatment of cancers. In this regard, he is involved in identifying gene expression profiles that predispose individuals to certain types of cancers. He has also developed novel methodologies for identifying kinases and phosphatases in biological samples. We are very glad and happy to have a scientist of eminence in our midst today to deliver a talk amidst the pandemic-related lockdown. His talk is entitled Use of Phycocyanin and Other Natural Products for Cancer Treatment. Dr. Appu Rathinavelu, you have 45 minutes from now to deliver your talk. Just 2 minutes before the end, I will remind you that you have 2 minutes left. After the talk is over, I will select a few questions from the chat box, the Q&A section, and will pose them to you. And the remaining questions will be mailed to you if we have to cut short the Q&A due to time reasons. Dr. Appu Rathinavelu.
Thank you, Dr. Suresh. Can you hear me well?
Yes, sir.
Thank you, sir. Thank you for a beautiful introduction. Also, I would like to thank Dr. Karthik Loganathan and other members of the organizing committee for inviting me to participate in this seminar symposium. It's a pleasure to be here. It's 7:15 a.m. here. I started joining this symposium around 6 o'clock, and I feel a little bit sleepy. So forgive me if I speak during the presentation. So I will try to share the screen and start with a brief introduction of our institution and then go from there. So let me share the screen. Are you able to see the slides? I hope you are. I'm not able to share the screen from here. So let me try again.
Sir, please open the PPT first, PowerPoint, and then share the screen, sir.
Yes, it's open. The PPT is open. So I'm trying to share through the Zoom.
Yes, the green share button and then share option.
Yes, it says one participant can share at a time. So I'm clicking on that, and then the screen is not coming up. I've done this many times, and unfortunately, I don't know what is happening now.
Just give me a minute, sir. I'll just check.
So while we are waiting for the screen to come up, maybe I can continue to talk. I am from NovaSoutheastern University, which is one of the private institutions in Florida, one of the largest private institutions. We have a total of 18 colleges, and within the health professions division, we have 7 colleges. Probably we are the only second university in the country, in the US, with 2 medical schools offering MD and DO programs. So we graduate nearly 3,000 doctors every year. I'm giving you these details to tell you, even though we are small, we are growing. The RumbaughGoodwin Institute for Cancer Research is one of the research units of Nova Southeastern University. We are engaged in cancer research. My research started, I would say, in '93, cancer research with NSU. And as Dr. Suresh mentioned, we have obtained not just 4 patents in the last 3 years, we have added 3 more patents. So we have 7 patents for small molecules, which are targeting angiogenesis. These are anti-angiogenic compounds. We are waiting for partners to collaborate with us so that we can do the clinical trials. So this is to show, let me try to share the screen again. It says 1 participant share the screen, and I'm unable to.
Sir, you must be able to share the screen now.
Now? Okay.
Please
so try again. Okay.
Yes.
So share the screen. Okay, it's not giving me the option to select the screen, unfortunately.
So click on the center of that share screen.
Share screen, yes, I'm clicking on that.
Click on the center of the share screen.
Okay, got it. Thank you for your, okay, now I got it. So I talked about the university. So can you see my slides now?
Yes, sir, we are able to see it.
Okay, all right, thank you so much. So this is one of the units, one of the centers of the Nova Southeastern University. And through innovative research, we want to develop new drugs and cure cancer. That's one of our goals. Recently I was looking at the number of COVID patients around the world. It's about 11 million. And if you look at the number of cancer cases around the world, it's 18.1 million. It grows, even though a couple of years ago, there was big news about decline in cancer incidence in the US. Hopefully we can see that in the rest of the world. When you go to MD Anderson Cancer Center, in most of the buildings, they have stricken out the name cancer, saying that we have conquered cancer. Now, in my opinion, we're not there yet. We will be there maybe in the next 10, 15 years, but conquering cancer is coming close to us, but we have to do our job to reach that goal. So as part of that effort, our institute is focusing on drug discovery, translational research, genomics, pathway analysis research, and we are also developing recombinant proteins that can target various cancers. And today's talk is going to be focusing on one of the natural products that we have tested in our research labs. We have tested the effects of resveratrol, genistein, curcumine, tulsi extracts, and also phycocyanin. Today's talk, I'm going to focus on phycocyanids. And as Dr. Suresh mentioned, we have several patents. Many of them are international patents, and we are in the process of now, starting the clinical trials in collaboration with the local companies. And if anybody from India wants to participate, we can really work together. Glioblastoma is one of our focuses because, this famous senator died because of glioblastoma, which created a lot of awareness. Since our compounds are targeting angiogenesis process, we are trying to use our compounds similar to Avastin for treating glioblastoma. It shows good brain delivery, and also Uma, who is participating in this seminar today, and also Dr. Tyagarajan, they are doing some of the analysis to confirm the effects of our compounds, F16 and JFD, for treating glioblastoma. So, and in addition to that, we are also looking at some of the side effects caused by chemotherapeutic agents. And that's one of the reasons why we are testing natural products. Basically, by using smart combinations, many of the drugs have come from plants and other living organisms, such as Taxol, topotecan, vincristine, vinblastine. But these are therapeutic compounds with a lot of pharmacological benefits. But our goal is to take the phytochemicals with nutritional value, antioxidant properties, and see whether we can really supplement the therapeutic effects of the chemotherapeutic agents. A lot of patients take supplements, dietary supplements, once the cancer starts. But our goal is to know the risks for developing cancer and start with these phytochemicals before the cancer starts and put the cancer on hold. And if the cancer starts, now use these phytochemicals in combination with the drugs such as topotecan, Taxol, and minimize the side effects. As you can see, peripheral neuropathy is one of the side effects that comes with chemotherapy. And our goal is to use combinations with the phytochemicals and minimize the side effects. This way, you are not only extending the therapeutic benefits and also minimizing any complications that may be coming along with the chemotherapy-induced neuropathy type of side effects. So, we are also doing drug discovery on the fast lane. Basically, as Dr. Ramanathan and other presenters were showing, they are doing a lot of docking studies, not myself, my collaborators. They are doing a lot of docking studies of the FDA approved, those drugs which are in clinical use, and see whether we can use them for treating cancers. By doing this, we are not only saving time, we are going to be saving a lot of money. For example, if it is taking 6 years, $300 million to bring a drug to clinical trials, by doing this repurposing, it is called repurposing, we can shorten the time and also save a lot of money in bringing an existing drug to patient's bedside. So this is one of our focuses. So some of the examples, for example, the arthritis drug, leflunomide, is used for treating prostate cancer. Dapsone can be used for treating Kaposi sarcoma. So these are some of the approaches we are utilizing at this point of time. Antioxidants are used not only by healthy individuals, and it is used by a lot of cancer patients because it can cause free radicals, can cause base modifications, and these base modifications can create effects similar to gene mutations and leading to cancer development. And our goal was to not only stop the production of the antioxidants by blocking the effect of the carcinogenic chemicals with the target tissues, and also minimize the process of induction of damages. As part of that effort, we are looking for phytochemicals from natural sources, antioxidants we are looking for from the natural sources, and we are looking for complex carbohydrates from natural sources. As one of the presenters, Dr. Tyagarajan mentioned, we are looking for omega-3 fatty acids from natural sources. All these are to maintain healthy life and also minimize the risk. Omega-3 fatty acids, I'm going to skip these slides for the benefit of time, are good in terms of providing support for maintaining healthy body. So one of our earliest studies were done with Tulsi extracts, and this should pave the foundation for the phycocyanin study I'm going to be presenting. Since a lot of people are coming into the waiting room, the screen is kind of coming up in the middle of the presentation, and I hope it won't be interfering in others' view. So when we looked at the effect of the Tulsi extract, our goal was to see whether Tulsi extract can systematically induce programmed cell death in cancer cells. And okay, hold on. Okay, so we selected LINCAP prostate cancer cells, and we did a dose response study. As you can see, the cells, when they were treated for 48 hours, we were able to see cell death with 100 microgram per ml of the ethanolic extract. 100 milligrams per 100 micrograms per ml is a kind of a heavy dose, but that was really for looking at the individual effect of the extract coming from the Tulsi. And we were able to establish a dose response curve for 24 to 48 hours. And we were able to see good effect in 48 hours. Of course, in 24 hours, there were some cell death, not to the significant level. And flow cytometry analysis also showed that this 100 microgram per ml of ethanolic extract can induce cell death. But that was not our original goal. Our original goal was to compare and also combine with drugs such as topotecan. For example, the level of cell death that was seen with 5 micromolar, as you can see, is very high compared to 100 microgram per ml of the ethanolic extract. But we were also able to do the fluorescence analysis. The propidium iodide binding study showed that both ethanolic extract and topotecan were able to induce cell death in these prostate cancer cell lines. So, and in addition to that, when we measured the caspase activation, both caspase 3 and caspase 9 activities were elevated with these treatments, and particularly with the ethanolic extract treatment. And that showed that the intrinsic pathway is activated. When you look at the apoptotic mechanisms, there are 2 pathways that are involved. One is the intrinsic pathway, the second one is the extrinsic pathway. This ethanolic extract coming from Tulsi seemed to be inducing the intrinsic pathway and leading to PARP cleavage, and that is what you are seeing here in this slide. In addition to cleaving the PARP, which is the DNA repair enzyme, in addition to cleaving PARP, the BCL2 level was also decreased by the ethanolic extract, which confirmed that both by decreasing the levels of BCL2 and also by activating the intrinsic pathway through elevation of caspase 3 and 9, caspase 9 and 3, Tulsi extract could induce cell death. So this kind of established our system for doing analysis with other compounds. This is when my collaborator, Dr. Miro Gantar, from one of the neighboring universities, came to me for collaboration. He was focusing on extracting phycocyanin from the blue-green algae that was growing in the Everglades. And I had a picture of the Everglades, but it looks like it is not appearing here. And if it appears, then I will talk about it for a few seconds. So this particular, let me close my screen 1 second. Okay, all right. So hopefully this, so this extract, the phycocyanin extract was, tested by Miro for other purposes, but he came to me for collaboration. And he wanted to, this is the Everglades. So the Everglades is very close to where we are in Florida, South Florida. And basically, I would say within a few minutes from where I live. And it has a lot of living, it's a natural habitat for a lot of living organisms. And, it has marshlands. And this is where the blue-green algae grows. And Miro was able to extract 2 strains of the blue-green algae, Limnothrix and spirulina, and he was able to extract phycocyanin. He wanted to see, even though there were many reports related to the effects of phycocyanin in stopping cell cycle progression and inducing cell death, he wanted to do systematic studies with our collaboration to determine whether phycocyanin can induce cell death in cancer cells. So as you can see here, phycocyanin, we prepared 1 micromolar solution, 10 micromolar solution. As expected, 10 micromolar was giving more than 60% cell death. I'm sorry, this is a topotecan. This one I was pointing at topotecan. So 10 micromolar topotecan was giving more than 60% cell death, but phycocyanin was not able to really show that much of cell death, which was not bad. But when we combined phycocyanin, the screen, let me minimize the screen. Okay, when we combined 500 microgram of phycocyanin with 1 micromolar topotecan, we were able to potentiate the effect of the phycocyanin. I'm sorry, the topotecan. So if you see the topotecan 1 micromolar, it was giving about 75% cell death. By combining with phycocyanin, we were able to really enhance the therapeutic benefit coming from topotecan. So the idea is by using combinations like this, we should be able to reduce the dose because higher dose produce side effects such as peripheral neuropathy. And by reducing the side effect, maintaining the therapeutic benefit, we would be able to treat the patient more successfully, and that they can continue with the treatment without going through the drug holidays. So this was the idea. And we were able to also look at the caspase activations, the combination, which is shown in the kind of the light purple bar, was able to show higher activities of the caspases. In addition to showing let me move the screen because this pop-up screen is blocking the blots. So as you can see that the BAX levels were completely elevated in combination treatments, and BCL2 was lowered. So, and cleaved caspase 3 activity or level was very much higher in the combination treatment. So these are individual treatments. As you can see, 500 microgram of phycocyanin did elevate the activities of the caspase 3, but the combination was working much better. This is with 1 micromolar topotecan, and of course with phycocyanin. So, this is a perfect example of something that can be taken as a dietary supplement if it is combined with right therapeutic agents such as topotecan, which is known to induce programmed cell death. It can, actually augment the effects of the topotecan by decreasing the levels of the BCL2 and elevating the levels of BAX and activating the intrinsic pathway. So let me move on to the next slide. So I'm trying to move on to the next slide, and it's kind of frozen. So let me see. Okay, all right. So part cleavage was also seen in these treatments, which confirmed that this combination can work in the prostate cancer cell line. And subsequently, we were testing, looking at the DNA fragmentation. This is one of the hallmarks of the apoptosis induction, where the DNA is fragmented, leading to completion of the cell death. So, at that point of time, Florida Department of Health released an announcement saying that, if we can use any of the natural products for treating lung cancers, we would be able to fund. So we started with the testing use the lung cancer cell line A55459 cell lines, and we were able to get some funding. When we looked at the gene expression profile by using the RT-PCR array, we were able to see elevation of the BAX in the array analysis also. In addition to that, we were able to see elevation of GAD45 and a few other genes, which confirmed that phycocyanin by itself can induce the expression of certain genes. And this was done with the lung cancer cell line, and the previous data I showed was for prostate cancer cell line. But the data you are seeing here is for the lung cancer cell line, which looked very similar to what we saw with the prostate cancer cell line. The combination of phycocyanin and topotecan 1 micromolar did the same thing. It was able to decrease the levels of the BCL2, elevated the levels of BAX, and that led to the cleavage of the PARP and completion of the cell death. So now the question was, this is a big molecule. Phycocyanin is a large molecule. I don't remember the molecular weight. Maybe Miro would be able to tell me the molecular weight immediately. It's a huge molecule. So we were wondering whether this molecule is able to really penetrate the cells. So this was a big question. And because this question was asked by many other reviewers, because huge molecules getting into the cells is not that easy unless you make pores by using detergents or some other alcoholic treatment. So when we did the binding study, fluorescence binding study, phycocyanin by nature, it can fluoresce. And when we were able to do the binding studies initially, after 1 wash, it was showing binding to the plasma membrane. As we were increasing the number of washes, after 2 washes, as you can see, the binding was decreasing, and after 3 washes, completely washed out. So that showed that the molecule is not entering, if the molecule is entering into the cells, it should be able to really stay inside of the cytoplasm. But what you see here is a dark field showing that whatever is bound to the plasma membrane got washed out. So that created the question, how would that be able to induce the cell death? So one of the targets we are focusing on is the FAS receptor. So we are suspecting that phycocyanin may be able to bind to the FAS receptor and activate what they call extrinsic pathway. And as you can see, that the combination of topotecan and phycocyanin was able to induce cell death, which was blocked by the FAS inhibitor. When we were using FAS inhibitor, for 3 hours, and then when we did the combination treatment, that inhibition was able to stop the effect, the extrinsic pathway activation by this combination, primarily by phycocyanin, was able to be blocked completely. So what is the role of topotecan? And the topotecan may be also able to induce extrinsic pathway to lead to cell death, and that was kind of blocked by the FAS inhibitor. So you can also see that the cleavage of PARP was inhibited by in the combination treatment by the FAS inhibitor. So this kind of indicated that it was not necessary for the whole phycocyanin to enter into the cells. Maybe by acting on the receptors, cell surface receptors, such as FAS receptor, it might be able to induce the extrinsic pathway through activation of caspase 8. It may be able to induce cell death, and that is what is shown in this diagram. So by activating the FAS receptor, it may be activating the caspases, and then it may be leading to activation of caspase 9 and 3 and leading to cytochrome C release, and which will eventually lead to cell death. And this is what we have confirmed when we treated xenograft animals, because once we completed the in vitro study, we wanted to see whether we are seeing any adverse drug effects in this during these combination treatments. And also we wanted to see whether this combination is working better than the monotherapies. And as you can see, phycocyanin, which was given in the drinking water to these xenograft animals, basically the A459 cell line was xenografted into these athymic mice, and we allowed for the tumor to develop for about a week. And after that, we treated them with phycocyanin in the drinking water, and whether it was Taxol or topotecan, it was injected intraperitoneally. And as you can see, that these drugs were able to really reduce the tumor burden. Phycocyanin was able to show some effect by itself. Phycocyanin was able to reduce the tumor burden, but when you let the animals to live longer than 60 days, in this group, phycocyanin group, the tumor started to grow more aggressively, and then eventually the animals died. In other two cases, that was not the case. That showed that phycocyanin can delay the growth of the cancer, not necessarily making the cancer to regress. So in terms of body weight, there were no significant changes in the body weight, which showed that there was some decrease in the Taxol, slight decreases in the Taxol and topotecan treatment up to 75 days. But with the phycocyanin, there was no significant changes in the body weight. And this is a picture showing the effects of the combinations. Here we showed how the combination with Taxol, phycocyanin, Taxol combination worked compared to the control. You can see that the tumor was growing more aggressively without any treatment. Phycocyanin was able to delay the growth. But when you combine that with Taxol or topotecan, we were able to see much significant therapeutic benefit. So this is kind of represented graphically in this picture. So basically what I'm trying to say is a lot of physicians are very hesitant to allow patients to take phytochemicals. Some of them, if they don't know about the benefits, they advise the patients not to take, in this case, for example, not to take any dietary supplements, because they may not know what kind of side effects it may produce. In order to take a safer approach, they recommend, tell us what you are taking. We will do some literature evaluation and then advise you whether you can continue to take it or not. But if we can do systematic studies, as we have done with ethanolic extract and with phycocyanin, we might be able to show to the scientific community that some of these phytochemicals can really work well with chemotherapeutic agents and provide the therapeutic benefits. So that was the original goal, and I think we have made significant contribution in showing to the world that this can be done. So let me go to the last 3 slides and introduce the rest of the team members here. So, we were able to get the funding from state of Florida for doing the lung cancer study, and Royal Dames of Cancer Research is one of our supporters. They are supporting our drug discovery research and other types of research that is ongoing at the RumbaughGoodwin Institute for Cancer Research. And we have recently received funding from the Community Foundation of Harvard for looking at the effects of anti-cancer drugs on peripheral nervous system for inducing peripheral neuropathy, because this is one of the common occurrences in multiple myeloma patients. When peripheral neuropathy develops, they have to stop the treatments and put the patients on drug holidays, which makes the cancer to progress very rapidly. And when we come up with the treatment measurements, such as treatment measures, such as use of phytochemicals or other combinations, we might be able to continue the treatment without putting the patient on hold. So this is one of our programs. So now we are training a lot of students, undergraduate medical students, and those who are sitting in the front row are undergraduate students, whether they are coming from US or from outside. We would be able to take them and train them during the summertime. Unfortunately, due to COVID situation, we were not able to accept any students at this time. And this is our team. All of these are graduate students on the left side, and Dr. Thiago, who might be online today listening to the presentation, and Dr. Minard, he is an expert in doing high throughput analysis. Dr. Umay, who's listening to the presentation, she's an expert in doing cell biology analysis. And these are the 3 graduate students who are with our team. And this is our vice president for operations, and he was visiting our institute recently, and we had a picture with him. And this is our team with all the members of the RGI. So I was going to show this video, but it may not work. And let me try, and looks like it is not going to work. So because of that, I'm going to go to the last slide and say thank you for your patience and listening to my presentation. If you have any questions, feel free to ask now. And I believe the moderators are going to receive the questions also and shoot the questions to me. Once again, thank you for your patience. Dr. Suresh, I'm not able to hear you because you are muted. There's a question in the chat. Yes.
Can you hear me?
Yes.
First of all, I'd like to congratulate Dr. Appu Rathinavelu for a wonderful talk. I think it was very informative, and I think the students can build on the fundamental knowledge that was taught to them by the teachers about the extrinsic and intrinsic pathways associated with cell death, and they can extend that to ethnopharmacology-based studies for drug development. Before I ask a few questions from my side, I thought I will give preference to the questions posed by the participants. First question is, does a particular phytochemical or combination, does a particular phytochemical or combination of 2 or more work for all types of cancer? Question posed by Shreya Churungu.
Dr. Shreya, good question. I wish I can say that there is a miracle phytochemical that is available that can cure all kinds of cancers. Not even chemotherapeutic agents that are currently available can cure all kinds of cancers. So we have to really see for which cancer this particular phytochemical is the most effective and try to use for that kind of cancer. And as you can see, phycocyanin worked well for both prostate and lung cancer. It may not work for other types of cancer, such as ovarian cancer or liver cancer, pancreatic cancer. So it would be, somebody was asking about customizing the medication. It would be a good idea to see for which cancer these phytochemicals are going to be the most effective and target those cancers.
Thank you, sir. The next question is, what cell would you use to study the effect of these phytochemicals in normal cells, and what will be those effects? Question posed by Bhagya Lakshmi.
Bhagyalakshmi, very nice question. We did use the normal epithelial cells I did not show the data. I believe it was in the presentation because the reviewers were very careful in asking what effects can you expect from phycocyanin on the normal cells, normal tissues. So, we did use the epithelial cells for testing the effects of phycocyanin. It was not really producing any significant side effects or toxicity. Very minimal cell death. Whatever cell death we observed were similar to what we typically see when the cells are growing without any treatment. So it's very safe. And the people, they, since this is a fluorescent, powder, they put it in drinks. And when you hit it under UV light, the drink fluoresces, and they love to drink those fluorescent drinks. That is how they consume in some of the parts of the world. The reason I'm mentioning about this is it's very safe. And when it is combined with Topotecan and Taxol, it was really potentiating the therapeutic benefits.
Okay, the next question is from Janatul Firdos. Is caspase-3 gene expression the only way to detect apoptosis? If it is not expressed, does it mean apoptosis did not take place when cell viability showed 60% death in the MTT assay?
Jana Natul, nice question. Caspase measurement is one of the methods for detecting apoptosis. It's a robust method. It's easy to do, but I would not rely on MTT because Uma, who's listening to this presentation, she was able to really look at cell death through 3 different mechanisms. One is apoptosis, second one is necroptosis, and she was able to really detect the markers for necroptosis and also necrosis. All these 3 can show decrease in the cell number if you're doing by MTT assay. Whether it is 60% or more, MTT assay is not going to be able to differentiate. If you're going to be looking for apoptosis, caspase activation is one of the measurements. We also use part cleavage. Not necessarily BCL2 depletion can be seen in all apoptotic mechanisms. Sometimes you may see Bax elevation without decrease in BCL2 levels, and because of that, you can do caspase activity measurements. You can do part cleavage, and this is what we typically do. And finally, DNA fragmentation to confirm. And if you have a good confocal microscope, you can see the fragmentation of the nucleus also. That is what Uma is going to be doing in the next few weeks. So these are some of the measures to make your reviewers excited when they are looking at your papers.
There's a question from a faculty, SBST faculty, Dr. Kavita Tirumurugan. What phase of the cell cycle is affected by phycocyanin?
Good question, Dr. Tirumurugan. Good question. We are thinking that it may be blocking the transition of the G1/S phase, but there's no confirmation yet, and we are going to be determining which phase is exactly working. But if you look at topotecan really blocks the G2 phase. That is a well-known fact. Some of the topoisomerase inhibitors, they block S phase also, but we believe that phycocyanin may be, kind of blocking the transition from G1 to S phase, but no, we haven't confirmed that yet. Very good question.
Thank you, Dr. Appu Rathinavelu. Now, question about glioblastoma from Jill.
Yes.
Have you performed any studies regarding ability of natural compounds to cross the blood-brain barrier? Thank you.
I'm waiting for somebody to give me a natural compound so that we can test it. So we have a very unique system. And the system we use is xenograft model, where we implant tumor into the brain. And then once we close it, it grows in about a week. It grows to a measurable size, because we cannot let the tumor to grow to 200 milligram, 300 milligrams. It'll cause a lot of cranial pressure. It will cause a lot of distress to the animals. So we have the imaging system which can detect when the tumor is established, measurable. So, and then we start the treatment. So this way, when we do the treatment, the drug has to go to the brain. It is not an intracranial injection. It is the IP injection or oral delivery, and the drug has to go to the brain and then stop the growth. And that is the system we have, which works very well. Actually, Mayo Clinic trained our scientist, Dr. Tiago, who may be listening to this presentation. He went to Mayo Clinic and learned the system, and that is what we have. If anybody wants to collaborate, tell me this is the compound we would like to test, and I would be more than happy to collaborate as long as there is some kind of funding available from somewhere. But so far, we have not tested the natural products because this xenograft system is very expensive, and without funding, without good goals, we don't want to really spend our resources.
A couple of questions one from Sheetal Rana. How effective would be personalized? How effective personalized medicines are for the treatment of cancer? Sheetal Rana.
Sheetal, I don't know whether you're a doctor or Dr. Rana or Sheetal. Good question. Personalized medicine is the way to go. And I'll give you one example, very quick example. Nowadays, as soon as they take out biopsy samples, they send it for genetic testing in the US. And they wait for the genetic testing to come out. Most of the profiling done nowadays are for cytochrome P450 system to make sure that the drug is not metabolized. And for example, if they are treating leukemia patients, they are looking for MGMT mutation polymorphism. MGMT polymorphism will determine whether the patient is going to tolerate the treatment or not. So basically nowadays the genetic testing is done for determining whether the patient is going to tolerate the treatment. If the patient is going to be tolerating high dose treatment, then they would be getting the treatments, the benefits. So that's one of the approaches. If they are not going to be tolerating the drug treatment because the enzyme is mutated or it's mutated means it's not going to be metabolizing the drug, it's going to be causing side effects, right? So in addition to that, they are looking for target mutations also. If a particular target is present, is it mutated? Is it going to be showing response for this drug? So that is the way to go. And I hope this is done in India in some of the hospitals, but the future of medicine is depending on how we can customize the treatment based on the genetic makeup. And because of that, I support that very much.
Dr. Appu, I just want to ask one question from my side.
Yes,
you're working on this Tulsi extract and topotecan combination, and then you went on to phycocyanin and the other combinations. What would be the next step in terms of improving the bioavailability?
Well, Dr. Suresh, we stopped studies with ethanolic extract and also with phycocyanin because of lack of funding. Getting funding from NIH for testing natural products is not very, very easy because there is, tons of literature available, and they feel that unless something that is very unique, they may not be able to, support the studies. We feel that our translational approach is very, very beneficial because if you dig into the literature, you may not see this kind of combination systematically looking at induction of apoptosis and other mechanisms that are related to cell death. But getting funding is the most difficult thing. And if the funding is available, we can certainly look at the bioavailability and other aspects. For example, in a phycocyanin, cyanobilin molecule can come out and it can go into the cells. And we haven't been able to do that study because of lack of funding. Right now, we are focusing on glioblastoma work because it's going to be attracting funding. We are focusing on peripheral neuropathy projects because we are going to be testing patients with that project. and that has given funding, and it's going to bring more funding in the future. So whatever is going to be benefiting the patients, and that is what NIH is going to be supporting. And if we can do the testing in India, it doesn't matter, whether it is bioavailability study or some other study with natural products, we can show that, and then we can attract funding.
Thank you, Dr. Rathinavelu. Before we end, one general question not related to cancer from Nirmal follows. Sir, could a natural product with immune boosting capability be helpful in treating coronavirus disease?
Actually, good question. Who was the general question?
Nirmal follows.
Nirmal Paulos, a very nice question, Nirmal. Actually, we submitted 2 proposals. Uma and Thiagu, they are co-investigators on those proposals, for treating COVID-19, one by using immunotherapeutic approach. What we are going to be doing is we are going to be creating a recombinant protein that will bind and neutralize COVID-19. And since this is going to resemble immunoglobulin, we are hoping that it can boost the immune system also. Now, of course, boosting the immune system is the best way to stop the multiplication of the virus. Even if the virus enters into the human body, which is becoming inevitable. If you look at the number of cases, not only in India and also in the US, one way or the other, virus is going to get into the body. And, our work should start from that point. We should be able to stop either by boosting the immune system or by developing therapeutics, which will be stopping the multiplication of the virus. From that perspective, I would say boosting the immune system is the best way. And this is the natural, process. And how we are going to be boosting the immune system? Are we going to be using any colony stimulating factors or some other therapeutic approaches? I don't know. This is what doctors have to test and come up with. But your point is very well taken, boosting the immune system. Are we going to be using natural products for boosting the immune system? At this point of time, I cannot think of any natural product that can boost the immune system to the level, the Nilavembu that was presented by one of the speakers, one of the junior faculty members, seemed to be used in many parts of the world. It seemed to be working because of the combination. So that is the approach, use of combination approach. Combination of natural products may boost the immune system and help.
Dr. Rathinavelu, can you take one more question, sir?
No problem, sir. I start my presentation well in advance for the questions, so I can take one more.
Okay, thank you, Professor. Aritra Ganguly, will FACS be effective for detecting cancer? What kind of mice is used? Is Swiss albino mice applicable for implantation of hypocyanin? What type of cancer grows in these mice? Can we observe asides in the mice?
Aritra, good questions. FACS can be effective for detecting cancer if you're going to be looking for a particular target. So it can be a gene that is mutated, and particularly if there is a mutant protein that you are going to be tagging, or if there is a way of detecting that protein by FACS. Basically by FACS, you're going to be isolating the cells, and you can do the second level of confirmation once those cells are sorted. So that is the approach. And actually, many of the clinics, we're going to have a flow cytometer for the purpose of doing cancer detection, but you need to have some kind of antibody or some kind of marking of the cancer-inducing protein or gene. So that is the way it is going to help. In terms of using mice, and Swiss albino mice can be used if you can suppress the immune system. Because if you're putting the xenografts are basically human tumors, or the tumors developed from the human cancer cell lines. When you put these xenografts into mice, most likely they're going to be rejected if the immune system is not suppressed. So it's necessary to use immunocompromised mice, or you have to suppress the immune system, then you can put any kind of tumor in these animals. But when you are suppressing the immune system using chemicals, that chemical may interfere with your study when you're looking at the anti-cancer properties. So you have to be very careful. There are a lot of immunocompromised mice available in India also. You should be able to use that for your studies. Did I answer your question?
Yeah, I think so. Thank you. On behalf of the organizers, I'd like to thank Dr. Appu Rathinavelu for a wonderful lecture. I think the students would have also been benefited from the results and also the experimental design. And some of the students would have been able to relate to the extrinsic and intrinsic pathways of cell death that are taught to them by their teachers. Thank you, Dr. Appu Rathinavelu. And I want to thank the organizers for having given me this opportunity to chair this session and listen to the wonderful lecture from Dr. Appu Rathinavelu. Over to Professor Jabez.
Professor Suresh, thank you very much, sir. Thank you for your patience and thank you for handling the session so well. And I also thank Dr. Appu Rathinavelu for his session. Thank you very much, sir, once more. So now we'll be moving on to the next session, which is the poster evaluation. So it starts right now from 5:30 onwards. So those who are presenting the posters can directly go and join the link and start your presentations. And if anybody else is having any other queries, I'll be here for another couple of minutes. So you can just post your question in the chat box so that I'll be there to help you out for just another 2 more minutes. Thank you very much. And tomorrow's session starts exactly at 10 a.m. And the first presenter speaker is Dr. Challa Suresh. So I request all of you to join by 9:45 itself sharp, so that the entire session will go on time. Thank you very much. Thank you all the participants. If you do not have any more questions, you're free to leave the day's talk session. Thank you.
Thank you.